Hello and welcome to another episode of EHA Unplugged. Today, we have with us Dr. Themeli and Dr. Ciprian Tomuleasa, who have extensive experience in CAR T-cell therapy. We've taken the opportunity to ask them a few questions. They provide an overview of the latest developments and novel ideas in CAR T therapy. Listen on and enjoy the podcast. Let's start with a brief introduction from our guests.
I'm Maria Themeli. I am assistant professor in the Department of Hematology in Amsterdam University Medical Centers. And I have a research line interest in developing novel CAR designs and off-the-shelf CAR T cells.
Yeah, so my name is Ciprian Tomuleasa. I'm from the School of Medicine and Inclusion in Transylvania in Romania. And I'm assistant professor in clinical hematology in the School of Medicine. And I coordinate a lab that is focused or is trying to focus on providing new potential targets or new alternatives for based on CAR T cells.
Could you start off our discussion by describing the current state of the art in CAR T treatments?
So at the moment, CAR T-cell therapies have been approved for hematological malignancies only, B-cell lymphomas and acute leukemias, lymphoblastic leukemias, and multiple myeloma. And we still have a big challenge in targeting solid tumors, and this is the holy grail of the next steps for CAR T-cell therapy research.
And right now in Romania, we have approved and reimbursed CAR T cells for B-cell acute lymphoblastic leukemia and also diffuse large B-cell lymphoma. It's not reimbursed for multiple myeloma. They're working in the health ministry on the reimbursement file, and all that process is going to be probably at the end of the year in 2023. And they do it right now under Dr. Tanase's coordination in Bucharest at Fundeni Institute. They did a lot, I think 10, 15 this year of patients with B-cell ALL.
What are the main challenges, limitation of the techniques, and the new innovations in the pipeline?
There are different levels for limitations. First of all, we see from the clinical trials, the clinical application of CAR T cells, that there's a specific percentage and a significant percentage of patients that relapse, so that CAR T cell therapy fails. And we're focusing on these patients in order to develop new versions of CAR T cell therapy that could also help them. Failures, as I said in my talk, have to do mostly with loss of the target antigen or downregulation of the target antigen. Also have to do with failure of the T cells to expand and kill the tumor. Another aspect is the immunosuppressive tumor microenvironment that, especially in solid tumors, needs to be addressed. And another challenge and another limitation has to do with the manufacturing of the CAR T cells. Oh, I forgot also the toxicities of CAR T cells. I forgot this is also very important. Off-target toxicities, CRS, ICANS, this is also a challenge. And then is the manufacturing process, which is lengthy, vein to vein time is up to 5 weeks sometimes. It costs a lot. And this is another point of improvement, which can be improved with novel ways of manufacturing, using different type of cells, different manufacturing methods, but also using allogeneic alternative sources for CAR T cells.
So, yeah, we have biological limitations related to the tumor biology of a malignancy, either a lymphoma or a myeloma or a B-cell ALL or etc. And also, we also have let's say, infrastructure limitations, because, not a lot of centers, not all transplant centers are accredited for CAR T cells, and not all of them should be, because we need very strict conditions in order to deliver this alternative to patient. And also, another important limitation is actually the price of the CAR T cells, the price of the CAR T cells offered by the industry. So that's why maybe academic design of CAR T cells and academic CARs might actually be a very good alternative in the future, in which they don't really have to cost like almost €500,000 per patient. So yeah.
And talking about innovation in CAR T cells, I think that the point of innovation right now will be the academic clinical trials. So it is very important that these academic trials are encouraged.
Speaking of trials, how close to being used are the latest innovations in CAR?
Now, CAR-NK cells have been used in clinical trials. There was MD Anderson is a leading institute in this aspect, using cord blood-derived NK cells transduced with a CAR, having important, significant results with using CD19 targeting. It's still not an approved therapy using CAR-NK cells. The limitation with CAR-NK cells is their persistence. For some malignancies, we may not need this persistence. For some others, we may. But the benefit, the advantage of NK cells is that they can be used as off-the-shelf therapy because they do not induce graft versus host disease. So to me, if you ask me, there is no optimal, the best source or the best cell type. I think for certain indications, CAR-NK may be better option. For other indications, CAR T cells may be a better option. And who knows, maybe in the future, you can have a mix of cell types, which maybe it's a very crazy idea, but why not?
Yes, theoretically you can transduce the CAR construct into not only any kind of immune cell, not only T cells. So CAR-NK cells might be an alternative therapeutic option. We just have to see the results of the phase 3 clinical trials, and afterwards, depending on the results of the trials, wait for the approval, and then we use them in the actual clinic.
What are some of the other innovations in CAR-T at the academic level?
The academic level innovations, I think double targeting, targeting 2 antigens instead of 1. I think this is the first thing that may come closer to a broader clinical application, because we see that the antigen loss is a quite important escape mechanism. But yes, a combination of co-stimulations to improve the effective function of CAR T cells. Addition, we heard now with CRISPR screens, we can identify transcription factors that their addition may improve the CAR T cell function. So the application of, in general, the application of CRISPR-Cas9 editing is another innovation that we expect to come into the field very soon. And I was very impressed by the innovations in the manufacturing field. All the efforts in order to shorten the time of manufacturing, improve the quality of the products, the amount, the yield of the products, using artificial intelligence in order to gather all the data and get all the information possible from all the data. So that was really, I think.
And also maybe a very interesting, maybe future hot topic might be the CARs that actually target the tumor microenvironment. Like we read the papers of the work of Dr. Themeli and Dr. Hudecek from Würzburg. They are working on CARs that actually target the tumor microenvironment folate receptor, so that it's still preclinical testing and still far away from a large clinical trial, but still, maybe in the medium future, this new alternative might find its place in the clinic.
And AML is then the next disease that we would like to target. And we heard yesterday and today about novel targets, novel combinatorial targeting systems to target AML, but also Dr. Carl June today presented data of efforts to be able, with CD45 targeting CARs, to target hematological malignancies in general, including AML. So I think that in the hematology field, AML is the next disease that we need to target.
Completely correct, because AML has a big unmet need. In B-cell ILL, for example, apart from CARs, we have alternatives like blinatumomab and other bispecific antibodies. But in AML, except for the small molecules like FLT3 positive AML, we have FLT3 inhibitors. But yeah, AML doesn't have a lot of alternatives in comparison with other malignancies.
Are there any other potential targets apart from AML?
If AML is targeted, I think almost all the most important hematological malignancies will be covered. The only thing that we need to find is additional targets, because we see that escape through antigen loss is important. So the more targets we have in our toolbox, the better. And going to solid tumors is a big challenge. And the solid tumors with the most unmet needs, like glioblastoma, so brain tumors, and the pancreatic tumors, and tumors with the highest incidence, breast cancer, lung cancer. I think these will be the most the focus areas for the next years.
Correct, because glioblastoma and pancreatic cancer have a very bad dismal prognosis, and 5-year survival rate is very, very low in comparison with other malignancies. So that is like a huge unmet need. Most of the patients diagnosed today with glioblastoma, stage 4 glioblastoma, will not be alive in 5 years. So yeah, they need alternatives apart from classic temozolomide and what they have today.
We hope you enjoyed this episode of EHA Unplugged. Check our show notes or visit the EHA Campus for more content related to CAR-T.